In the last lecture, we spent a lot of time talking about cloning genes by positional cloning. And this is a good approach, but there are problems associated with things like positional cloning. And one of the biggest ones is that you often don't have large enough populations to do positional cloning, or at least you have a very difficult time doing positional cloning due to limitations in population size. I'll talk a little bit about why that is in a second. One of the other problems in identifying genes that are associated either with diseases or other phenotypes that you might be interested in is that several genes contribute to the phenotype, and this is very common. So I want to talk a little bit today about some of the potential solutions to this. And in particular, we're going to talk about linkage disequilibrium mapping and quantitative trait loci mapping. Both of these things are fairly complicated to carry out, but I think the basic genetic ideas are actually not all that difficult, and I think we can cover those fairly easily. So positional cloning, so this is just a figure from our last lecture. One of the big limitations is the number of individuals that you have in your mapping population. In this example, we have a lot of offspring that are affected with this disease, but as you can imagine, this is probably even a little optimistic that you would get 7 individuals out of the same family that would have the disease that you're interested in. And this is a big limitation. So numbers are a real problem, and if you can kind of think about this for a bit, so what you're really getting is information from the number of recombinants between the disease-causing locus and whatever markers you're using. So the resolution is really dependent on how many recombinants you can find. So for instance, if you have only 10 individuals, then if you find one recombination event between a marker and the locus of interest, then that would indicate that one recombinant out of 10, that this would be about 10 centimorgans away. And this is quite a ways. So this is about 10 million base pairs in a rabbit or in humans, and there would be hundreds of genes in that interval. And you can kind of see that the steps that you would take in terms of defining this interval are very large with this number of individuals. So one recombinant would be about 10 centimorgans. If you sort of went up by one recombinant and you detected 2 recombination events out of these 10, then that's 20 centimorgans. So we're kind of taking these giant steps across the chromosome, mainly because we have so few individuals. So if you were lucky, or if you work in a model system where it's fairly easy to get large numbers of individuals, if you were looking at 1,000 individuals, the resolution with which you can map the position of genes is much higher. So if you had one recombinant out of these 1,000 individuals, then that would be about 0.1 centimorgans, or 100 times closer than here. And similarly, if you move then up by one recombinant, so instead of having one recombinant out of 1,000, you have 2 recombinants out of 1,000, that only moves you up to 0.2 centimorgans. So with a larger population, you can move across the chromosome in much smaller intervals and give you a much better idea of where these genes lie. And the problem is getting 1,000 individuals in a human population is pretty tough. So one clever solution to this is something called linkage disequilibrium mapping. So this is another way to identify the positions of genes on chromosomes, looking for markers that are associated with phenotypes. Only it takes advantage of naturally existing populations. So again, experimentally this is very difficult to do, but conceptually I think it's fairly easy to understand why this works. So this is an approach which overcomes that limitation of the number of individuals in pedigrees. So an example of how you can do linkage disequilibrium mapping deals with Iceland. So linkage disequilibrium mapping works particularly well if you have a large group of individuals that are all distantly related to each other. And Iceland works very well for that. So Iceland was founded by a few hundred Vikings about 1,000 years ago. So they started with a very small number of people, and now there's a few 100,000 people that live in Iceland. And because of the fact that not too many people move in or out of Iceland, not too many people go to Iceland, not too many people leave Iceland, most of the people that currently exist in Iceland are descendants of those Vikings. So when you think about that first population that founded Iceland a few hundred, or about 1,000 years ago, there were a few hundred individuals. And some of those individuals will carry genetic diseases, just like anybody does. And most will be carriers, so most of them won't actually be showing the diseases, they won't be homozygous, but they'll just be carriers. So let's just look at one example. Let's say we have one original Viking, we're going to call him Sven, that carries a mutation for albinism. So he's just a carrier. He's not homozygous, he's not an albino, he's just a carrier. But let's say that Sven is also the only founder that carries this mutation for albinism. So again, there's only a few hundred people. The idea that only one of them would carry a particular mutation in a particular gene is not an unreasonable one. So what this means then is that everyone else is homozygous wild type for this gene that would cause albinism, and Sven is heterozygous. He carries this one mutation. So what does this do for us? Well, Iceland now, 1,000 years later, has about 275,000 people. Almost all are descended from those original few hundred Vikings. So let's say now that there are maybe 500 individuals in Iceland that are albinos. So where did these mutations come from that are causing this albinism in these individuals? Well, they could be new mutations that have occurred in the population, but those occur fairly rarely, so there's only about 10 to the minus ninth mutations per round of DNA replication. So probably not. Also, not many people move to Iceland, so what that means is that most likely all 500 individuals are homozygous for the allele that Sven was carrying 1,000 years ago. And this has some pretty important implications. What it means is that if you were to actually sequence the genomes and sequence this gene from all the wild-type individuals in Iceland and all the albinos in Iceland, all the wild-type individuals would have what we're calling the wild-type sequence, and all the albinos would carry the exact same mutation, the one that Sven had 1,000 years ago. So what this means then is, if we could actually just sequence everybody in the population, then everybody who's normal would have this wild-type sequence, all the albinos would be homozygous for Sven's allele. So how does this help us? Well, we have to introduce something called a haplotype now. So we've talked a lot about genotypes, and haplotypes are basically just unique combinations of polymorphisms that are together on the chromosome. And because they're together on the chromosome, they tend to get inherited together, because it's not too likely to have recombination events occurring in between these closely linked sets of polymorphisms. So what I've drawn here in these colors is on the black chromosome is a set of polymorphisms that are going to be associated with this particular chromosome, and a set of polymorphisms which in red are associated with this chromosome. So if we think about Sven's original allele, so Sven was heterozygous, he had one chromosome he inherited, say, from his mother in black and one from his father in red. During meiosis, whenever Sven is producing sperm and getting ready to pass his genes on to the next generation, there's a chance for recombination to happen, so we can have these sort of recombinant alleles taking place, and then these get passed on to the next generation. So, whenever one of Sven's offspring has children, so again, this would be like Sven's original chromosome that was passed on to his offspring, this offspring then is going to mate with another individual that's going to produce yet a different set of haplotypes, which I've done in blue. And they mate, and again there's a chance for recombination to happen, so we can shuffle these alleles some more, and these get passed on again to the next generation. And what's important to realize, though, is that these haplotypes, so these collections of polymorphisms that are close to this mutation which is causing albinisms, will tend to get inherited together, just because they're very close together on the chromosome, so it's less likely to have a recombination event that's going to separate this mutation from these closely linked polymorphisms. So what this means is that even after 40 generations or so, in those 1,000 years, is that if you look at all those chromosomes that carry this mutation of Sven's, they're all going to have this mutation which causes albinism, but by and large, as you look at the other polymorphisms on the chromosome that are close by, they will tend to get co-inherited also. So along with this polymorphism that's actually causing the disease, these single nucleotide polymorphisms or microsatellite polymorphisms, which are close by, will also get inherited. But as you get further away, then there's going to be more of a chance for recombination to have come in and separated these, and I tried to show that all these different haplotypes and these different polymorphisms by all these different colors. So, in individuals that carry Sven's mutant allele, nearby they're also going to all have the same polymorphisms that all came from Sven's original chromosome. Whereas if you look at wild-type individuals in the population, they will just be assorting randomly for this particular position. So we haven't selected them out for any reason. So the individuals who have the disease, have albinism, are all going to be homozygous for this and will contain all these linked mutations, or linked polymorphisms as well, whereas a control group in the population who aren't carrying that allele will just segregate randomly. So this is largely the basis of linkage disequilibrium mapping. So all of the individuals that have the disease have inherited the same allele, so the same chromosome from that same ancestor. Then they should all have the same mutation in the same gene, along with all of the other linked polymorphisms, like the single nucleotide polymorphisms, which occur very frequently in the genome. So again, there's only about 25,000 genes in the genome, but an individual carries about 3 million single nucleotide polymorphisms. So there's a lot more of these polymorphisms than there are genes. And those SNPs that are very tightly linked to the mutation causing the phenotype will tend to get inherited together, even after 40 generations. So what this means is that if you were to scan along the chromosome, then the region containing the mutation in that gene that's causing our disease, albinism, should also contain all the same single nucleotide polymorphisms, i.e. they should have the same haplotype. So this only really works very well if you can be confident that most of the individuals with the disease would have inherited it from a common ancestor. So there's only certain populations that this really works very well for. So Iceland works very well. There are also some other populations which also tend to be fairly isolated. Sometimes these can be physically isolated, like in Iceland, where they're just on an island that's isolated from other individuals, or you can end up with populations that are isolated from the others, from other groups. Isolated means that they don't reproduce, they don't mate with others because of something like religious beliefs. So the Amish in parts of Pennsylvania and Ohio and Indiana typically do not end up marrying or mating with outsiders. So it's another example where there's a small group that tends to breed within their own group. So if you want to do single linkage disequilibrium mapping, one of the difficulties with it is that you need to look at a lot of single nucleotide polymorphisms. So you need to look at a very large number of markers all along the chromosome to identify these sorts of patterns, where all the individuals which have the disease are going to have the same markers, where unrelated ones will have different markers. Luckily, with microarrays, this is fairly easy to do this type of allele-specific oligohybridization, which we talked about a couple of lectures ago. So you can literally look at tens of thousands or hundreds of thousands of single nucleotide polymorphisms at a time. So this gives you a very good look at lots and lots of markers across the chromosomes. And basically then what you're looking for is what I mentioned before. So at locations along the chromosome that don't have anything to do with that particular mutation, or in individuals that don't carry it, these single nucleotide polymorphisms are just going to be randomly distributed across the genome, or across the population. So you're not going to see any particular correlation with this locus with the disease. But when you get to a locus where that mutation lies, you're going to go from seeing essentially a random assortment of polymorphisms in these individuals to, as you get closer and closer to that mutation that's actually causing the phenotype, you're going to see more and more of those single nucleotide polymorphisms that are just like the ones that Sven had. So when you get to the point where everybody is showing the same polymorphisms, this tells you that you're very close to finding this gene. Now this has been a powerful technique, especially with Iceland. So Iceland realized that it was kind of sitting on a genetic gold mine, in part because of the way their population had evolved, starting from a very small number of Vikings 1,000 years ago to these 275, 300,000 people now. And not only is it good for those reasons, but also because Icelanders are very good at keeping their pedigrees, keeping their family histories, and they actually formed a company to essentially handle their genetic material, which is called Decode Genetics. And over half of the entire population of Iceland has participated. They've been willing to donate their genetic material, their samples of their DNA to be sequenced, and also all of the history from genealogical studies and also genetic and personal medical information. And already this has been quite successful, and they've cloned probably a few dozen now, but as of a few years ago they had 12 genes associated with various diseases that were all able to be cloned by linkage disequilibrium mapping. So what's in it for Iceland? So why would they want to donate all their material for this? Basically, the deal is that any drug therapies or drug screenings that result from using their DNA, that they would get for free for Iceland. So this is a good deal for Iceland because their entire country is going to get free medical screening and drugs and therapies from the result from these studies. And for companies, this is not really too big of a deal because Iceland is not a very large market, so they can still make tons of money selling these drugs to the rest of the world. So it seems like a fairly reasonable deal. Okay, for the last couple of minutes of this lecture, I want to tell you a little bit about the analysis of complex traits. And these are traits that arise from the interaction of two or more genes. And we talked a little bit about this fairly early on in the course. And this complicates a lot of genetic analyses. And unfortunately, most traits probably fall into these categories. So things like height, weight, eye color, hair color, muscle mass, longevity, your predisposition to things like diabetes or osteoporosis or cancer, all are going to be the results of multiple genes. And this makes identifying, mapping, and ultimately cloning these genes much more difficult. So one of the approaches to go after these sorts of genes when there's going to be more than one gene involved is something called quantitative trait analysis, or QTL mapping. And this is a basic workhorse technique that's used in all sorts of studies, especially in natural populations and agriculture, but also in human studies as well. I picked an agricultural example because I think it's one of the easiest to follow. So the idea with QTL mapping is you're dealing with traits that are complicated, or complex, that they're being controlled by more than one gene. But you would still like to know where are those genes and how can we identify these genes. So the example we're talking about deals with pulp content in tomatoes. So these ones would be considered to have too little pulp and they're getting kind of watery and squishy, and these ones have a better pulp concentration. So if you wanted to understand the genetic basis of pulp content, this is kind of an idea of how you would do this. So if you end up with inbred lines, so these are pure breeding lines, or homozygous at all loci, so the whole genomes are essentially homozygous. And we have one of these lines which has very low pulp content, and one that has very high pulp content. And the idea is that if we do a cross between these two, we should be able to see the effect of whatever genes are causing the high pulp content here, and whichever genes are causing the low pulp content here. We should see these segregate in an F2. So that's what we set up here. So these parents, one has low pulp, one has high pulp, cross them together, we get an F1, cross those together, we get an F2. And in this F2 then, we can look at relative pulp content. And you can see that it ranges the whole way from something which was close to the original high pulp parent, so at about 99% pulp content, down to very, very low pulp content, like maybe 10%. And almost everything in between. So if you were to plot these out and try to look at the distribution as to whether you can see an easy segregation amongst these pulp contents, you probably can't. You're not going to see 3-to-1 segregation because it's not just one gene. It might be 2, might be 3, might be 5, might be 10. So how do we go about identifying genes out of this mess then? So it's a lot like quantitative, or a lot like linkage disequilibrium mapping, in that the key is to look at lots of markers across the chromosomes. Now, in this situation, we dealt with a low pulp parent and a high pulp parent. And again, if you think about those chromosomes, both of these parents were homozygous to start with, so I'm kind of drawing the low pulp chromosomes in blue and the high pulp ones in black. And then again, we put in lots of markers. So these could be single nucleotide polymorphisms or just any type of polymorphisms that you can look at across the chromosomes. So in the F1, then, there is a chance for these things to recombine. So in the F2, you're going to end up with some loci that are homozygous for the low pulp parent, some are homozygous for the high pulp parent, and many of them are going to be heterozygous. And then some of these lines are going to have low pulp content, some will have intermediate, some will have high. And then the basic idea is to just scan along the chromosome and ask, do you see any correlation between the genotype and the pulp content? So if you see a particular position where all of the individuals which have high pulp content in the F2 also have these black polymorphisms, so the polymorphisms that came from the high pulp content parent, then that could tell you that that's a region which is associated with high pulp content, that there's a gene involved in there that you've detected. And similarly, if you look and you notice that the ones with lower pulp content tend to be associated with one particular genotype, that could also give you another idea that there's a region that's involved in this. So the math of this is fairly complicated, but the concept, I think, isn't so terrible. You're basically just going along the chromosome and asking, do these markers correlate with the genotype? Do the markers correlate the genotype with the pulp content for that particular position on the chromosome? So what this actually looks like then in a map is something like this. So correlation between genotype and pulp content, and then position on the chromosome. So again, we're trying lots of markers along the chromosome, and sometimes there'll be no correlation whatsoever. So it doesn't matter if your DNA at this particular position came from the high pulp content, came from the low pulp content parent. It doesn't make any difference with regards to what sort of a phenotype you're seeing. But then as you get to other positions on the chromosome, you'll find that there is a correlation. That say most of the plants in the F2 that have high pulp content had single nucleotide polymorphisms that came from the high pulp content parent. And this tells you that there must be some gene in this region that's contributing to this phenotype. But again, because this is not a single gene trait, you may find other positions on the chromosome that also correlate between the genotype at this position and the pulp content. So these are the sorts of things that you'll see in quantitative trait analysis, or positions on the chromosome that are contributing to the phenotype. And some of them may contribute more than others. So this would be a gene that has a stronger effect than this one does. So in this way, we can kind of take apart complicated traits which are determined by multiple genes and map those positions onto the genome.